Hello dear student, today's lab will be about identification of bacterial pathogen. The identification of bacterial pathogen is a cornerstone of medical microbiology and a critical component of clinical diagnosis and patient management. Accurate identification allows clinicians to determine the causative agent of infectious disease, select appropriate antimicrobial therapy, prevent complication. Bacterial identification is not a single test, but a systemic step-by-step process that integrates clinical information with laboratory finding. This process begins with proper specimen collection and handling, followed by microscopic examination, culture on appropriate media, and a series of biochemical, immunological, and molecular tests. Now let's have an overview on what medical microbiology is and medical microbiologist. Medical microbiology is a specialized branch of medical science that focuses on the study of microorganisms that are not visible to the naked eye, including bacteria, viruses, fungi, and parasites. This field emphasizes understanding their biological characteristics, mechanism of pathogenicity, and their direct impact on human health and diseases. The medical microbiologist is a person who plays a crucial role in the diagnostic laboratory by accurately detecting and identifying infectious agents. This role is essential for correct diagnosis, appropriate treatment selection, prevention of disease transmission, and effective infection control, particularly in hospital and community settings. Since our lab is about identification of bacterial pathogens, let's see what they are. What are pathogenic bacteria? Pathogenic bacteria are harmful bacteria that can cause disease to human, animal, and plant. But remember, not all bacteria are harmful. Many are useful, but pathogenic bacteria cause illness. Now let's see, where do they come from? Pathogenic bacteria can be found in many places, such as air, breathing when we breathe, coughing or sneezing. In water, the water we drink, it could be contaminated. The food we eat, uncooked or contaminated food, they exist in soil, animals, human. This is why hygiene and sanitation are very important. How do laboratorians deal with pathogenic bacteria? Clinical laboratory can grow bacteria, isolate them, and identify them, know exactly which bacteria it is. And why identification is important? Identification is important to diagnose the disease correctly, choose the right antibiotic, prevent spread of infection, and save life. There are many methods that we can use to identify this kind of microorganism: the phenotypic, immunological, and genotypic. But the method that we are focusing today is the phenotypic one, the morphology, which we are going to culture this kind of microorganisms. Phenotypic method, what it looks like, this method is based on appearance and behaviors. We are going to look at the shape of the bacteria, size, color of colonies, how it looks under the microscope, how it grows on culture media. Just by recognizing person by their face or height, bacteria can be recognized by how they look. So today we are going to do the culturing for bacteria. In this slide, we are going to talk about how can we be successful in identification of microorganisms. Let's see what they are depends on. We have the aseptic technique. Aseptic technique means working cleanly and safely, wearing gloves, using sterile tools, avoiding contamination from hands, air, or surfaces. To make sure only the bacteria from the patient is tested, not other germs. and correct obtaining the specimen. The specimen is very important in this process because we need to obtain it very carefully without any contamination. A specimen is a sample taken from the patient, such as blood, urine, sputum, swab from throat or wound. If the wrong sample is taken, the bacteria cannot be found. And correctly handling the specimen. After collection, the sample must be handled carefully, kept in the correct container, not shaken or spilled, stored at the right temperature, and quickly transporting the specimen to the lab for diagnosis, for testing. Time is very important. Samples should reach the lab as fast as possible. Delay can cause bacteria to die or multiply wrongly. And culturing and identification the bacteria in the lab. In the lab, the specimen is placed on culture media. Bacteria are allowed to grow, and we are going to identify the bacteria using tests. This step helps doctor know what bacteria is causing disease and choose the correct treatment. Okay, now let's get to the culturing of bacteria. It is a process of placing bacteria on special food so that they can grow. Bacteria are very small and cannot be seen easily. When we grow them, they form colonies that we can observe and test. What is the culture media? Culture media is the food for bacteria. Okay, it can be solid or liquid, like broth. The culture media that we are going to use today is a solid culture media, like agar plates. The requirement that we are going to use today in our lab is this sample. We are going to have two samples, the blood sample and urine sample. And then we are going to have gloves, inoculation lobe, culture media, Bunsen burner, and incubator. And those two bottles that you're going to see are blood culturing bottles. Before we culture blood, we are going to test them whether the bacteria present inside this blood or not. And we are going to do that by putting blood into those blood culture bottles and placing them into a back alert for, I think it's for 5 to 6 days until we would be sure if the bacteria exists in the blood, and then we are going to culture that blood. As we said before, we need to test the blood sample whether the bacteria exists or not, because sometimes instead of bacteria there's fungi in the blood. So the process of blood sample, as we said before, we're going to put them in the blood bottles, culture bottles, drawing blood from patient. A small amount of blood is taken from the patient using a sterile needle. Then putting blood into culture bottles. The blood is placed into two special bottles. We have two special bottles, the aerobic bottles and anaerobic bottles. Aerobic bottles for bacteria that need oxygen, but anaerobic bottles for bacteria that grows without oxygen. Why two bottles? Because different bacteria grow in different conditions. And then we load the bottle into the analyzer. It is an automated analyzer which called Bactalert. The machine keeps the bottle warm. It checks continuously for bacterial growth and detection and result. If the bacteria grows, the machine shows a positive result. Lab staff know bacteria are present. So, after the presence of bacteria has been confirmed, we are going to culture them. Now let's look at the common media used in microbiology lab. Culture media are essential tools in medical microbiology because they provide the nutrients and conditions required for bacterial growth, allowing isolation, observation, and identification of pathogens. Each medium is designed for a specific diagnosis purpose. The first one, we have the nutrient agar, a basic, non-selective, and non-differential medium. What do we mean by non-selective? It means it's a general nutrient agar for every microorganism. All bacteria can grow on this nutrient agar, support the growth of non-fastidious bacteria, used for routine cultivation, observing colony morphology, size, shape, and color, and do not distinguish between different bacteria. As we said before, it's a non-selective. The second one, we have McConkey agar. McConkey agar is a selective. See? Nutrient agar, non-selective. McConkey agar is selective and differential media. Selective meaning only one type of bacteria can grow on this media and inhibit the other type of bacteria. McConkey agar is selective for gram-negative bacteria, which means only gram-negative bacteria can grow on this kind of agar. And it's differential based on lactose fermentation. It has a lactose fermenter. Pink colonies, for example, the E. coli, and non-lactose fermenter. Colorless colony, for example, Salmonella. The third one is blood agar. Blood agar enriched and differential media. It contains 5% blood, support fastidious organisms, differentiate bacteria based on hemolysis. We have 3 types of hemolysis: alpha, beta, and gamma. When the growth is in alpha hemolysis, it's a partial and greenish. Beta hemolysis, it's complete hemolysis and clear zone. Gamma hemolysis, it's none. And the fourth one is chocolate agars. It's also an enriched and non-selective media. Released growth factor like X and V factors used for fastidious organisms such as Haemophilus influenza and Neisseria species. And then we have the mannitol salt agar. It's a selective and differential media. High salt concentration, it's about 7.5% select for Staphylococci. Differential based on mannitol fermentation. Fermenter, yellow media, Staphylococcus aureus. And non-fermenter, pink media. And the last one is Eucine methylene blue agar. It's also selective and differential media. Selective for gram-negative bacteria only. Differential for lactose fermentation, exactly like McConkey agar. And then we have the culture techniques. There are 4 methods for culturing bacteria. But the method that we are going to use today is streaking method, to get separate colonies in order to obtain pure culture and commonly used for isolation and bacterial from clinical specimen. The other are spreading method, stabbing method, and stabbing and streaking method. Spreading method to obtain carpet growth for drug sensitivity test. Stabbing, it's used for motility test. Motility means we are going to see if the bacteria is motile or non-motile in semi-solid media. Stabbing and streaking for studying biochemical tests in both slant and butts. The streaking method is a basic microbiology technique used on solid media, agar plates, to separate bacteria so that we can obtain pure colonies that come from a single bacterial cell. Why is this important, if you ask me? Pure colonies are needed to identify bacteria, study their characteristics, and perform antibiotic sensitivity tests. The general idea of streaking, a small amount of bacteria sample is placed on an agar plate. The sample is spread in a specific pattern using a sterile lobe. Each streak dilutes the bacteria more and more. By the last streak, individual bacteria are separated and grow into a single colony. There are types of streaking method. We have the interrupted streaking. The plate is streaked in separate sections. The lobe is lifted between streaks. This reduces the number of bacteria in each section. It helps in gradual dilution and isolation of colonies. And as you can see, it's a interrupted streaking method we used on this culture media. Now let's look at the step-by-step interrupted streaking process. At first, we're going to sterilize our lobe by heating it with a Bunsen burner. And then we are going to dip the lobe in the bacterial sample. Streaking is done in one small area of the plate. This area has the highest number of bacteria to place the original bacteria onto the agar. And then we are going to streak it in a small area on the other side, as you can see in the figure A, B, C, D. This is how interrupted streaking is being done. After completing streaking or culturing, the agar plates must be incubated inverted, upside down. This step is very important for obtaining accurate and pure result. Why do we incubate plates inverted? To prevent condensation. During incubation, water vapor forms inside the plate. If the plate is kept upright, water droplets collect on the lid. These droplets can fall onto the agar surface and to maintain isolated colonies. When water drops fall on the agar, bacteria from one colony can spread to other areas. Colonies may merge. And to ensure accurate identification, inverted incubation keeps colony separated and well defined, allows correct observation of colony size, shape, and color. At the end, we are going to mention some aim of culturing bacteria. As we said before, it's a method of multiplying microbial organisms by letting them reproduce in prepared culture media under controlled laboratory conditions. Microbial culture are used to determine the type of organism, obtain pure culture, do subsequent microbiological testing. It is one of the primary diagnostic methods of microbiology. To be able to study the cultural, morphological, and physiological characteristics of any individual microbial species, used for antibiotic sensitivity tests, and used to make stock cultures.